Good morning, everyone, and thank you for the organizers for having me. Well, my name is Veliz-Otani. I work at the University of Maryland, Baltimore. I'm a last year PhD student working with Timothy O'Connor. And there's been a little change, using the wrong mouse. There's been a little change in that title of the presentation. I hope that's not a big problem. So the title of the presentation I will show you now is Unifying Population Structure and Relatedness Analysis through a Coalescent Approach, with a big asterisk on coalescent. And well, first of all, what is relatedness? It's simply a measure of genetic similarity, usually in terms of identity by descent, and there are many ways to measure relatedness. Two of the most common ways are the kinship coefficient, which is the probability that two alleles are IVD, two allele samples from different individuals are IVD, and the coefficient of relationship that is twice the kinship coefficient. And this is what usually the genetic relationship matrices that we use in GWAS will try to estimate. And why do we care about relatedness? Is because this is what allows us to model the phenotypic covariance between relatives. However, the traditional definitions of relatedness are in terms of identity by descent. And as we know, in these traditional definitions, we assume that beyond a baseline generation, there's no more similarity between any individual. So all the chromosomes in these baseline generations are different from each other. So we cannot measure in this framework more similarity beyond this baseline. Of course, now we have genomic approaches where we can estimate IVD from genomic data, and we don't need to define baseline generation, but still we are limited to a few dozen maybe generations by identity by descent analysis. And from a coalescent theory point of view, as we know, everyone's related. So if we expanded this pedigree back in time, eventually everyone will coalesce to a common ancestor. And this is sometimes called or partitioned into recent relatedness and distant relatedness, where recent relatedness basically refers to familial relationships measured through IVD sharing, or through scale genotype covariances, such as the genetic relationship matrices. And the distant relatedness refers to shared ancestry measured through principal components analysis. I will skip this because of time. So I will talk about some genotype-based estimators of the kinship coefficient and their limitations, which is basically the motivation for the method I will show them. So one of the most common methods is GCTA, which GCTA is the name of the software, which, well, doesn't matter what it stands for. But basically it's an approximation of the kinship coefficient as a scale genotype covariances. If X is the genotype, so we have the indices I and J for individuals and K for sites, then we basically have the center, the genotype centered at the mean, well, the product of them divided by the variance under Hardy-Weinberg equilibrium. And then we divide by the number of markers, and that will be an estimator of twice the kinship coefficient. However, this measure does not account for population structure or admixture. The RIP method is an extension or a modification of the GCTA method that does account for population structure. It basically assumes that there are ancestry-specific allele frequencies, and that there are also subject-specific ancestry proportions. So from this, we can estimate what would be the expected allele frequency of an individual given its ancestry proportions and the allele frequencies in each ancestry. So from this, we replace the previous allele frequencies we had, and we get an estimator of the kinship coefficient that accounts for population structure. However, this requires a correct specification of the ancestry components, the proportions, and the allele frequencies in each ancestry. A further and final extension that I will mention is PC-relate, which drops the need to define the latent components. And instead, we simply regress the genotypes on the principal components. So we'll have the expected genotypes given the ancestry of a sample, and we will get a similar estimator of the kinship coefficient that actually works really well, even in highly admixed samples. Another generalization of the GCTA matrix, but from a different perspective, is this. So this is, again, the GCTA model, but now in matrix form. It's basically the cross product of the standardized genotype matrices divided by the number of sites, where the standardized genotype for an individual at a given site K is just the mean-centered genotype divided by standard deviation. The LDAK model, which is an important extension to address different genetic architectures, assumes that this scaling factor could have different values. So basically, it introduces an alpha parameter that if we set it to -1, will reduce to the GCTA model, but we can basically let it be whatever value, or we can try to fit it to the data and infer the alpha parameter for different traits. And the reason why this is important is because then if we assume that on the scaled genotype space, all the genetic variants have the same effect or have the same variance, then in the raw genotype scale, the effect sizes will have a variance that depends on the allele frequency. For example, for -1, which is the default value in GCTA, we will have that rare variants have a higher effect on the phenotype, which is sometimes considered a proxy for natural selection. And with the alpha parameter, we can change how much more weight we give to rare variants. Now, the downsides of these methods is that first, RIP and PC relate intentionally remove information because they only want or they only attempt to estimate recent relatedness. And GCTA and LDAK are considered biased because they are not good estimators of the kinship coefficient when there is population structure, but arguably they have more information, even though their interpretation is not so clear. So the research question then that I present is, can we measure a relatedness back to the most recent common ancestor? And can we use it to model the phenotype covariance in mixed linear models? And this is the coefficient of genealogical similarity that I developed that I will present. So first, we need to consider the full genealogy of a set of haplotypes as a series of local trees, where each tree represents the genealogy for a genomic region. And then because of recombination, we'll have different trees along the genome. So first, let's focus on a single tree. We'll have 3 time parameters for each pair of haplotypes. We have the time to the root of the tree, and we'll have the shared divergence time and the unshared divergence time. The shared divergence time is the time during which any mutation will be shared by both haplotypes, and the unshared divergence time will be the time during which any mutation will be not shared by the haplotypes. And then we can see the genotypes of 2 diploid individuals as just a diplotype, so a pair of haplotypes per individual, and we will come back to this in a moment. So we now need to specify a phenotype model, and we will express the phenotype as deviations from the ancestral phenotype y sub 0. These curly braces IJ represent the diplotype of an individual, in this case with haplotypes I and J. So this subject will deviate from the ancestral phenotype a certain amount that depends on all the mutations carried by both haplotypes, the deviations attributed to haplotype I and deviations attributed to haplotype J, plus some random variation. And similarly for the other individuals within these haplotypes H and K. So we want first to estimate the covariance between these phenotype values, because the y sub 0 is a constant, and we assume that there is no covariance between the random error. The covariance between the phenotypes will be the covariance between the random deviations due to genetic effects. So we want this, the covariance between these terms. So we will assume an additive model, and we'll define DI for any haplotype I, where DI is the deviation due to the mutations in that haplotype, which will be the sum of all the effects of the mutations carried by that haplotype. So beta sub m is the effect of the mutation M, where M goes from 1 to n sub I, where n sub I is the number of mutations in that haplotype. If we assume that the number of mutations in a haplotype follows a Poisson distribution with a parameter that depends on the mutation rate, the sequence length, and the time during which we are following that haplotype, then if the effect sizes follow some distribution, which we do not need to specify, we just need to assume that the variance does exist, then the variance of the total deviations will be given by this parameter here. So basically, we have the Poisson lambda parameter and the time, and the moments of the effect sizes. Now we have the variance, but what we want is the covariance. First, we need to notice that we can decompose the deviations, the phenotypic deviations, into the shared deviations, which are the deviations caused by the shared mutations, and the unshared deviations, which are the deviations caused by unshared mutations. Then the covariance, then we can again repeat the sum, but for different time frame. And the covariance between di and dj will be the covariance between these sums. But if we assume that the effect, the phenotype deviations that occur here are uncorrelated to the mutations that happen here, because the mutational process become independent once they diverge, then there will be no correlation between the effect sizes that accumulate here or here. And similarly, there's no correlation between the effect sizes that happen here and here or here. I think I should change to that later. This makes things better. Where was I? So one thing that I have mentioned is that I think implicitly we're assuming that there's no selection, because if there was selection and all the haplotypes that are being selected for the same environment, possibly there could be a correlation between the deviations accumulated. But if we assume there's no selection, then we can assume there's no correlation. Then the covariance between these terms will be simply the covariance between these two terms. And this becomes the variance of the deviations during the shared time. And because we derived previously the variance of the deviations, we now know its form. The only difference is now that the time is not the time from present to the root, it's just the shared time. So that was for the deviations of two haplotypes, but now we need the deviation for the total phenotype of two diploid individuals. We proceed in a similar way, but now we'll have two haplotypes per individual. So we will need to estimate the covariance between each term. And after some algebra, it will reduce to this expression, where basically the covariance between the phenotypic deviations depends on all the pairwise shared time. However, we have these parameters that we don't know, we cannot easily estimate. So to get rid of them, we can estimate the correlation. And if we estimate the correlation, it reduces and we'll lose all those parameters. And then we can express the correlation of the phenotypic deviation from the ancestral phenotype simply as a ratio of the shared time parameters. And this is what I define as the coefficient of genealogical similarity for two individuals. In this case, now I'm changing the indices to A and B to refer to two diploid individuals that carry the diplotypes IJ and HK. And something I haven't mentioned here is that actually this expression has two formal definitions. It's the correlation of the number of derived alleles carried by two diploid individuals, but it's also, and this is the proof that I just showed, is the correlation of phenotype deviations from the ancestral phenotype under specific genetic architectures, which was that the variance is the same for all the mutations, right? Which would correspond in the LDAK model to an alpha of 0, which is different from GCTA, which assumes alpha equal -1. To use this coefficient in a mixed linear model, we would need to make some assumptions, but basically if we want to model the covariance between the genetic effects, then it will be the estimate of Jesse times the estimate of the genetic variance. But in this case, we will not have a single, actually, a true parameter of the genetic variance, but the genetic variance will be some sort of the average of the product of the standard deviations of all potential pairs in the sample. And this is largely because the variance, if we remember, I should have repeated it, is not the same for all individuals. I don't have it here. I didn't include it here, but basically depends on the amount of autozygosity, which is not the same for every individual. And that's why we cannot assume that there's a single variance for the whole population or sample, but it's specific for each individual. So the estimate of the genetic variance will be an average of all the products of the standard deviations. Now the question is, can we use it in practice? So I did some simulations using MS Prime. I simulated some data. Some data using MS Prime from using this model. I plotted this model from I made this figure from this model by Medina Munoz 2023, which is a demographic model of Latino and Latin American populations, where basically we have anatomically modern humans that then split into two components, Yoruba, representative of African ancestry and one out of Africa. And then we have these other populations, which are Puerto Ricans, Yoruba in Ibadan, Nigeria, Chinese in Beijing, Iberians in Spain, Peruvians in Lima, Mexicans in Los Angeles, Colombians in Medellin, and indigenous Americans from Mexico, which is an inferred or a theoretical component. And then each of these components will be, admixed components will be the result of these other ancestry donors. So I simulated, first I started with a very small simulation because it was a pilot to validate the model. Well, these are the details, but basically we have 60 samples per population. And then when I plot the GC coefficient for all the samples, we see that a clear clustering that represents the labels. And so it represents the population labels, but we see that its distribution also depends on the levels of familial relationship. But what is important here and what is different from the kinship coefficient is that if we notice here the yellow violin, the value for unrelated individuals is not the same in all populations. For example, in Chinese in Beijing and in indigenous Americans from Mexico, the similarity is higher in unrelated individuals than in populations from Africa, because as we know, there's much more diversity in Africa, and that is being represented here, which is something that we cannot represent with a kinship coefficient. So yes, it's reflecting both distant and recent relatedness. However, most of the time we do not know the true genealogy of a sample, and it's very hard to estimate, especially when we have a mixture, and it's also very computationally demanding. But I derive a genotype-based estimator. 3 minutes, okay, I will try to run. Basically, the genotype-based estimator I prove is that the cosine similarity of the genotype vectors represents, is an estimator of the GC coefficient based on the time parameters. And the reason why this is important is because if this is a cosine similarity, then if we take the arccosine, we get an angle, and then we can interpret that angle as a measure of distance. And then we can run a principal coordinates analysis and use it to also detect population structure. But first, let's see how good of an estimator is this genotype-based estimator. We see that the correlation is very high between the true tree data and the genotype-based estimator. And then when I compare the principal coordinates analysis, the composition of the basic coefficients to the conventional PCA, we see a better separation of these samples from the Peruvian Genome Project, plus some samples from public data sets. Now, to be completely transparent, it didn't always perform so well compared to PCA. Most of the time, performed similarly. In this particular case, it performed much better than traditional PCA. Some caveats is that this initial definition of GC measures the covariance of the phenotypes with respect to the ancestral phenotype, which of course we don't know. We will want to know we will want to measure the covariance as deviations from the mean. Ideally, the population mean. However, most of the time it's hard to define what the population is in GWAS samples. So I will use instead the sample mean. And we assume that the effect size of all mutations follow the same distribution because we assume alpha equals 0. However, we can generalize it. And if I have time, I will present that. No. Okay. But basically, just to mention what I meant by deviation from the mean, basically, this is what we are modeling with GC, the variance created by time, the phenotypic variance created by time. But in a real sample, we do not have that. We have the variance only of the ascertained samples. So we want to measure the covariance as deviation from the mean of these ascertained samples. So what I did is a geometric extension, which is not an analytical proof, but then I demonstrated that it works well empirically with a bunch of simulations. But basically, if the cosine similarity of the genotype vectors measured as deviations from the ancestral sequence is an estimator of the Pearson correlation of the phenotype deviation from the ancestral phenotype, then we can shift the reference to the sample mean in both the cosine similarity and the Pearson correlation. And sorry, you will have to wait for the paper.
10 seconds.
So basically, it works well.
That was really cool. Thank you. Could you give me some intuition as to how your tree-based estimator is different from like an expected GRM given an arc?
Yes, so I think you're talking from that paper from maybe 2023. Yes, so that paper, they proved that their inference target is actually equal to the GCTA when you use the whole genome sequence data, if I remember correctly. This estimator is very similar, and it's actually the standardized version of the GCTA matrix. So basically, if you calculate GCTA from whole genome sequence data and standardize it by dividing it by the cross product of its diagonal, you get this estimator. That's the relationship.
So you said you want to account for a population structure in your model, but I see like you still using like homogeneous populations. We are doing simulation. So I don't know if you have any like future directions on like testing this like trust your model on like a 2-way or 3-way mixed populations.
So these populations were much more advanced than that because I had 8 populations, and each of these that I chose Latin American populations because in different countries we'll have a higher, in some countries, higher proportion of indigenous Americans, in other higher proportion of Europeans, and in other populations higher proportion of African ancestry. And it's very heterogeneous across countries. But what I did in other simulations that I didn't have time to show is then I introduced artificially an additional admixture event 3 generations ago, so that every individual could have, 2 generations ago, so that every individual could have a grandparent coming from different generations. And then I used also that, and it was a bigger sample size too. Yeah, and it perfectly works well.
Yeah, I was wondering how sensitive your estimator would be to non-genetic deviations that would point in the same direction.
I did some simulations of introducing confounding that is correlated to population labels. And something I couldn't mention is that I also tested the need to include principal components, because the idea is that if the matrix is capturing any level of population structure, then in theory, you don't need the principal components to account for the ancestry itself, unless there's confounding that is correlated specifically with those principal components. So when you know the true value of alpha, using the principal components in this case, which was modeled using the population labels, which I think reflects better confounding, because confounding will not be truly linear with the amount of ancestry in any axis. It's very artificial to think that. So in this case, using principal components did not help. It did not make things worse either, but it didn't contribute anything.